Have you ever wondered how the genetic code actually works in our bodies? I mean, when you hear the word code, what do you think of? Maybe people hunched over tables, scribbling away, working with secret codes and encryptions. Maybe you think of computer security and authentication and so forth. Either way, that doesn't really help you envision what's going on in our bodies, in all of our cells. So what's going on? In this video, I'm going to show you exactly how this works. So first, the DNA double helix everyone is used to. The key here is it consists of 4 nucleotides: adenine, thymine, guanine, and cytosine. We just abbreviate these as A, T, G, and C. They fit together, but the adenine only fits with the thymine, and the guanine only fits with the cytosine. So if you look at that double helix, wherever you see an adenine or an A, it's going to have a T in the opposing coil. Wherever you see a G, it'll have a C in the opposing coil when things are working properly. That's the way it's going to be. This is the important base pairing rules that DNA follows. It's important to know that. G goes with C, A goes with T. We'll see why this is important in a minute. Okay, so here's a little more detailed look at how they fit together. The T and the A have 2 hydrogen bonds. The G and the C have 3 hydrogen bonds. That's the base pairing. Okay, now the first step, if you're going to use the genetic code, you have to have a message. And the message is a copy of the DNA. This big purple machine here rides along the DNA and makes a copy. The copy is shown in red. The copy is called messenger RNA or mRNA. So it's just a copy of one of the 2 coils of the DNA. One little detail, the thymine or T is now replaced with uracil or U. Okay, the base pairing still works the same way. It's just you're now using a U instead of a T. Okay, now it's going to get edited. The mRNA is going to get edited and then passed on to the ribosome for the translation step. Here's the translation step. You see that mRNA molecule there running along the bottom. It's got those nucleotides, A, C, G, and U. And then there are these other machines here, these smaller molecules called transfer or tRNAs. They have 3 nucleotides sticking out, and they will mate up with the mRNA 3 at a time. So you see a couple of them there in the ribosome mated up. Those nucleotides sticking out of the tRNAs are pairing up with the mRNA nucleotides using those same base pairing rules we just looked at. That's why I emphasized those same base pairing rules are being used here at the ribosome. And here you see another tRNA getting ready to mate up. That one has the 3 nucleotides UUU, which will mate up with AAA here in the mRNA strand. Now at the other end of the tRNAs is an amino acid. Here it's lysine. The earlier one had phenylalanine and methionine. These amino acids will be used by the ribosome to build the new protein. It will grab these one at a time and stitch them together, and you'll have a string of amino acids. So that's why they call this the translation step, because you're translating a string of nucleotides into a string of amino acids. That string of amino acids will fold up into a protein eventually. Now you have a problem here in that when these tRNAs are used, they leave without an amino acid. They can no longer fulfill that role. They are depleted. They're spent. They're uncharged. They need to be recharged. There are special proteins that are going to do that. Here's an example, special protein, a cartoon view of a protein with an amino acid attached to it. Now it's going to look around for a spent tRNA. Look, it's found a spent tRNA. It's going to mate up with this spent tRNA and attach the amino acid to it. Here's an illustration. The tRNA then leaves with the amino acid. So this special protein has attached this amino acid to the tRNA. Okay? The key here is that the correct amino acid needs to be attached to the tRNA. There's an army of these proteins, and they carry one of the 20 amino acids, and they're looking for the correct tRNA to match it up to. They are the implementers of this genetic code, okay? They have to get the right amino acid attached to the right tRNA. There's an army of these proteins. They carry the different amino acids, and they're looking for the different tRNAs. They get the right one, the right amino acid attached to the right tRNA. The result is The special proteins attach the correct amino acid to the correct tRNA, and you have the familiar genetic code, this table showing the 64 different triplets of nucleotides and their corresponding amino acids. The secret sauce is all in those special proteins that get the right amino acid attached to the right tRNAs. If that didn't happen, none of this would work. And this whole system is used to make proteins. It's called cellular protein synthesis. The genetic code is at the heart of it. It's used to create proteins. Of course, proteins are required for the whole system, and that secret sauce is an army of proteins. You need the pre-existence of proteins to make proteins.